We start first topic for the next colloquium. We will have 4 topics and then colloquium on them. In this block of topics we will discuss digestive system and then respiratory system and the skin. For this colloquium you will have both test and oral part. We will start with the digestive system, exactly supradiaphragmatic part. Digestive system includes gastrointestinal tract, which is the tube, including oral cavity, esophagus, stomach, small intestine, large intestine. And also the large glands are included in digestive system. These are 3 pairs of salivary glands which produce saliva, liver which produces bile, and pancreas which produces enzymes for digestion process. Today we will discuss the supradiaphragmatic part of the gastrointestinal tract. It means that we will discuss the organs of oral cavity and esophagus. Everything which is about the diaphragm, which is located somewhere here. Gastrointestinal tract has 3 subdivisions: anterior portion, which includes oral cavity, pharynx, and esophagus. Then middle portion, which includes stomach, small intestine, and large intestine, and colon. And posterior portion, which includes the caudal portion of the rectum. So today we discuss just anterior portion. The functions of gastrointestinal tract include the function of motility, movement of the food, secretion, production of some secretions such as saliva or enzymes, digestion, which means breaking down of food particles and molecules to smaller ones, and absorption, which means transport of the products of digestion into the cells of the epithelium of the gastrointestinal tract and then further into the blood. So, in oral cavity and esophagus, the function of motility includes mastication, which means chewing, swallowing, mixing, and propulsion of the bolus. Bolus is the mass of food which was chewed, masticated with the teeth, and mixed with saliva. Then this portion of food is called bolus and it travels along the gastrointestinal tract. The function of secretion in oral cavity includes the production of saliva, the fluid which fills our oral cavity, also enzymes, because the saliva contains some enzymes mainly for digestion of carbohydrates, and hormones are also produced by some cells in the oral cavity. The function of digestion here is the digestion of carbohydrates, sugars, just initial steps. And absorption is almost absent in oral cavity and esophagus, but in the area of the floor of the oral cavity, just below the tongue, we have a lot of blood vessels, and here we can have some, for some extent, the absorption, and this is one of the routes of administration of medicine drugs. It is called sublingual. For example, some pills for the heart attack are used in such a way that they are placed just below the tongue. And there, these pills are absorbed very fast because of the presence of very high amount of blood vessels, and the saliva serves as the dissolvent. Esophagus just makes the way for the food, which is called bolus now, from the oral cavity towards the stomach. So in esophagus, nothing is digested and absorbed. Food just travels towards the stomach. Here, the function of motility is produced and secretion of the mucus secret, which lubricates the surface of the esophagus and helps the food, the bolus, to move downwards towards the stomach. In the stomach, we have the peristalsis, which includes mixing and propulsion of food. Also, the food is stored here for up to 6 or 10 hours, depending on the type of food. For example, the food rich in proteins will be stored for longer time in stomach. The secretion here is the secretion of hydrochloric acid, which makes here very low pH level, enzymes, which serve for breaking down of proteins, mucus, and hormones are also produced here in stomach. Function of digestion. Here, the digestion of proteins, fats, and carbohydrates starts. So these organic molecules start to be broken down into smaller pieces, but absorption will take part next in small intestine. In small intestine, function of motility includes segmentation and peristalsis, mixing and propulsion of bolus. Secretion of bicarbonates, which neutralize the pH level of the food, because the food which came from stomach has very low pH level, and it can damage the epithelium of the small and large intestine. So when the food comes from stomach, the pH level should be neutralized with the help of bicarbonate ions. Enzymes in small intestine mainly come from pancreas. Also, bile comes into small intestine from the liver and gallbladder. Mucus is produced here in small intestine, and hormones. Digestion. Here in the small intestine, most of all the organic molecules are absorbed and digested, such as proteins, fats, and carbohydrates. And these large molecules were broken down into monomers, peptides were broken into amino acids, carbohydrates into glucose and fructose, lipids into smaller ones, and all these small molecules can now be absorbed into the epithelium of the small intestine. Also, water, ions, minerals, and vitamins are absorbed here. In large intestine, the colon, the function of motility is provided. It's also the mixing and propulsion of the bolus. Secretion here, the production of mucus and some hormones. No enzymes are produced here. And the function of digestion here is called bacterial digestion, because here in large intestine, in colon, we have about 2 kilograms of symbiotic bacteria. They always live in our large intestine, and they help us to digest those molecules that could not be digested earlier previously in small intestine because of lack of some enzymes. So these molecules can be then digested, broken down into smaller molecules with the help of the bacterial enzymes, and this is called bacterial digestion. Here in large intestine, mainly water, ions, minerals, and vitamins are absorbed. And then here the bolus becomes more hard and turns into feces. Embryonic development of the gastrointestinal tract is shown in these pictures. Here you can see this process in the sagittal section of the embryo, and here in the frontal section. As a result of cephalocaudal folding shown here, cephalocaudal folding, and the lateral folding shown here. So as a result of this, we have the closed primitive gut, shown in yellow color. This is the epithelium of the gut, future intestine. Here it is shown also in yellow color. This is the primitive gut, and it is separated from the outer environment with the cloacal membrane and oropharyngeal membrane. The black line here shows the ectoderm, which in the frontal and in the caudal portion of the embryo forms invagination towards the oropharyngeal and cloacal membrane, and will form the oral cavity in future and the distal portion of the rectum. Further, the oropharyngeal and cloacal membranes will rupture, and the definitive gut of the embryo will be opened on both sides. So as the result, we have such sources of development of gastrointestinal tract. They differ mainly in the part of the epithelium. So in the oral cavity and in caudal portion of rectum, the embryonic source of development of the epithelium is ectoderm in both cases. In esophagus, the embryonic sources of development of the epithelium is precaudal plate. And most of the length of the gastrointestinal tract, which includes the epithelium of stomach and small and large intestine, here the epithelium derives from the endoderm. This is only about the epithelium, and also the structure of epithelium changes. It is stratified squamous, non-keratinized or partly keratinized in oral cavity and esophagus, and also in the caudal portion of the rectum. In stomach and small and large intestine, we have simple columnar epithelium. So mainly epithelium changes along the length of the gastrointestinal tract. Other layers of the wall of the gastrointestinal tract include such tissues such as connective tissue, muscular elements, blood vessel, nerval tissue, neural plexuses. They all derive from mesenchyme. And in the oral cavity, this is the neuromesenchyme because it derives from the neural crest. The organs of gastrointestinal tract, which include stomach, small and large intestine, are also covered with mesothelium because they are located in the abdominal cavity, and all organs which are located in some cavities are covered with mesothelium. So the stomach, large and small intestine are, so they are covered with mesothelium, and mesothelium derives from the visceral layer of the splanchtome, or other name is the splanchnic layer of the lateral plate mesoderm. Now, the gastrointestinal tract wall has 4 layers. We start always from the inside. The innermost layer is the tunica mucosa. It has 3 sublayers: tunica epithelia, or just epithelium, lamina propria mucosa, which contains loose fibrous connective tissue, and in some cases simple glands. Lymphoid follicles can be also present. And lamina muscularis mucosa, which contains smooth muscle tissue. Second layer is called tunica submucosa. It contains loose fibrous connective tissue with blood vessels, muscular arteries and veins, also arterioles and venous and capillaries, neural elements such as Meissner or submucosal nerve plexus. Nerve plexus includes intramural ganglia and nerves which connect those ganglia. Also in submucosa, the glands can be present, and if they are present there, they are always compound. Third layer is called tunica muscularis or muscularis externa. Usually it contains two layers, but in the stomach it contains three layers. And the type of muscle tissue is different in different parts of gastrointestinal tract. Above the diaphragm, in the organs of the oral cavity, in pharynx and in esophagus, we have striated muscle tissue because it is voluntary and it helps us to chew and swallow the food voluntary when we want to. Below the diaphragm, the tunica muscularis contains smooth muscle tissue. Usually, the orientation of the layers of muscularis are inner layer circular, concentric, and outer longitudinal. There may be some differences in different parts of gastrointestinal tract. Between the layers, between the inner and outer layer, we have loose connective tissue with the nerve plexus, which is called Auerbach's nerve plexus or myenteric, and also blood and lymphatic vessels. The outer layer of the gastrointestinal tract wall is the tunica externa, which can be adventitia or serosa. Above the diaphragm, for example, in esophagus, we have adventitia, which contains loose fibrous connective tissue. Below the diaphragm, we have serosa, which contains not only loose fibrous connective tissue, but also mesothelium, which covers it from the outside. Here you see the same, but in the picture. Four layers are shown in this example of small or large intestine. So the innermost layer is the mucosa. It includes epithelium, this white line, then lamina propria mucosa, yellow in this picture. This is loose fibrous connective tissue with these ball-shaped lymphoid follicles, and lamina muscularis mucosa, red line. Next tunica is the tunica submucosa, here, blue in this picture. It is loose fibrous connective tissue with blood vessels and nerve structures. Here you see the nerve plexus, which is the Meissner plexus. It includes ganglia and nerve fibers. The glands, if they are present in the tunica submucosa, they are always compound. Next goes the tunica muscularis, which usually contains two layers, inner circular and outer longitudinal. Between those layers we have thin layer of loose fibrous connective tissue with the elements of nerve plexus, Auerbach's nerve plexus, and also blood vessels. The outer layer can be adventitia or serosa. In the case of small intestine, this is the serosa, because the small intestine is covered from outside with mesothelium. In this picture, you see the difference in the histological structure between different portions of the gastrointestinal tract. In the first picture A, we see esophagus, in B, stomach, in C, small intestine, in D, large intestine or colon. So, in all cases, we have four layers. The tunica mucosa, then tunica submucosa, then muscularis externa, and the outer layer, which can be adventitia in esophagus, and in all other layers, in all other organs, serosa. So all four layers are present everywhere. Above the diaphragm, as you can see here, we have the stratified epithelium and the adventitia. Below the diaphragm we have simple one-layered epithelium derived from endoderm, and the outer layer is the serosa. Tunica mucosa includes three lamina or sublayers. The innermost, which is the closest to the lumen, is called lamina epithelia or just epithelium. In oral cavity and esophagus, the epithelium is stratified, squamous, non-keratinized or partly keratinized in some areas of the oral cavity, and the source of development for epithelium of oral cavity is ectoderm, in esophagus precaudal plate. In stomach, the epithelium is simple columnar, glandular epithelium. In small intestine, we have the simple columnar epithelium with brush border, and in large intestine, just simple columnar epithelium. As you can see here, epithelium can lie flat or form some invaginations. For example, in stomach, epithelium has these pits and glands. In small intestine, the epithelium forms both evaginations, where epithelium covers the villi, and invaginations in crypts. In large intestine, we have just invaginations, crypts. Lamina propria is loose fibrous connective tissue just below the basement membrane of epithelium. So, for example, here in stomach, everywhere just below the basement membrane of epithelium, we have loose fibrous connective tissue of lamina propria. It is rich in blood vessels, nerves, and the elements of immune system. Lamina muscularis is shown here. It usually contains 2 or 3 layers of muscle elements. It always contains smooth muscle cells, so here you see the smooth muscle cells, and the number of layers of the lamina muscularis and the orientation of smooth muscles is the same as in the muscularis externa. But the function of the lamina muscularis mucosa is not the propulsion of food, but is to change and maintain the shape of the tunica mucosa. If tunica mucosa forms some folds or some other elements of the changing shape, so it is because of the lamina muscularis. The difference between parts of alimentary canal or gastrointestinal tract are those: the relief of the inner surface. Relief means the shape of the surface. The surface can be flat or it can form some evaginations or invaginations. So it will be called relief. Second, type of epithelium and its source of development. Third, the presence and location of the glands. Fourth, the number of muscular layers in the tunica muscularis and the type of muscle tissue in it. And is it adventitia or serosa? So, first, type of relief, the shape of the surface, of the inner surface. In tongue, the relief will include the lingual papillae. Here you see these evaginations, which are the evaginations of the lamina propria mucosa and the epithelium. In esophagus, we have longitudinal folds, which are seen in anatomical preparations. The fold, which is shown here, is composed of both tunica mucosa and tunica submucosa. This is the largest element of relief, and it is made by 2 tunics, mucosa and submucosa. And it is usually can be seen in naked eye without microscope, like the longitudinal folds in esophagus. In stomach, we have the folds. You can see them here. They are large, so they are seen with naked eye. And the smaller elements of relief, gastric fields and gastric pits. Gastric pits are seen here. They are small invaginations of the epithelium into the lamina propria mucosa. In small intestine, we have also folds seen in anatomical preparation and microscopic elements of the relief such as villi and crypts. Villi are evaginations of epithelium and lamina propria, and the crypts are invaginations of the epithelium into the lamina propria mucosa. In large intestine, we also have folds and crypts. No villi, just crypts. Second, type of epithelium and its embryonic source of development. In oral cavity, stratified squamous keratinized, or in some areas non-keratinized, derived from ectoderm. In esophagus, simple columnar stratified squamous non-keratinized, derived from precaudal plate. In stomach, simple columnar mucus-producing epithelium derived from endoderm. In small intestine, simple columnar absorptive epithelium with brush border with microvilli derived from endoderm. In large intestine, simple columnar epithelium derived from endoderm. In rectum, not shown here, stratified squamous non-keratinized from ectoderm. Next, the presence and location of glands. In the organs of oral cavity, the salivary glands are present in lips, shown in this picture, so this is the location of the secretory portions of the salivary glands. Also in tongue, within the muscular body in submucosa and mucosa of the ventral side, on the hard palate glandular zone in submucosa, and also in soft palate, where we have the mucous glands in submucosa. In esophagus, shown in this picture, we have two kinds of glands. The large mucus-producing glands, which are called esophageal glands proper, the major type of the glands of esophagus. They are located in the submucosa. And the second type, minor type of glands, the cardiac glands. They are simple tubular and they are located in the mucosa. The border between the mucosa and submucosa is usually the tunica muscularis mucosa. It is the outer part of the mucosa because mucosa contains epithelium, lamina propria, and lamina muscularis. So where muscle elements of lamina muscularis and then starts the submucosa. So to find the border between mucosa and submucosa is to find the tunica muscularis mucosa, lamina muscularis mucosa. In stomach, the glands are called gastric glands, and they are located in mucosa, in lamina propria mucosa, above the lamina muscularis. In small intestine, glands are present only in duodenum, because the small intestine contains 3 intestines. Just after the stomach goes the duodenum, then next jejunum and ileum. So the glands are present only in duodenum. It is shown in this picture. So the first part is for duodenum, and here we have the compound glands located in the submucosa. In next portions of the small intestine, in jejunum and ileum, we have no glands. In large intestine, no glands are present. Next, the number of muscle layers in the muscularis and the type of muscle tissue in it. In the esophagus, we have two layers, the inner circular and outer longitudinal. But the type of muscle tissue differs when we go along the esophagus from the pharynx towards the stomach. So, if we subdivide the esophagus into three parts, the upper, the middle, and the lower part, in the upper 1/3 of the esophagus, we have only skeletal muscle tissue in the muscularis externa. It is shown in this picture here. In the outer layer, we have the longitudinal sections of muscle fibers. In the inner circular muscular layer, we have the cross sections of muscle fibers. In the middle 1/3 of the length of the esophagus, we have both skeletal muscle tissue, which contains muscle fibers, and smooth muscle tissue, which includes smooth muscle cells. This is the area where the switch of the type of muscle tissue takes place. Here, the skeletal muscle tissue changes and is substituted with smooth muscle tissue. And in the lower portion of the esophagus, closer to the stomach, we have only smooth muscle tissue in both inner circular and outer longitudinal layer. In stomach, we have three layers of smooth muscle tissue: outer longitudinal, then middle circular, and additional the innermost oblique layer of smooth muscle tissue. Because the stomach is the large muscular organ, it needs a lot of muscles to perform the storage and the propulsion and mixing of the food, so it needs additional layer of the muscle tissue. You can see here how different muscle tissue look like in histological preparation. Here you can see longitudinal sections of the muscle fibers of skeletal muscle tissue, and here cross sections of muscle fibers with the nuclei of them located at the periphery. And here you can see both longitudinal and cross sections of smooth muscle cells in smooth muscle tissue. So they are spindle-shaped on the longitudinal section and look like small circles in cross-section. In small and large intestine, we have two layers, outer longitudinal and inner circular layers of muscularis externa, which contains just smooth muscle tissue. And adventitia or serosa above the diaphragm. In the esophagus, we have the adventitia, and in the area where the esophagus goes through the diaphragm to reach the stomach, it becomes covered with the serosa, like all organs below the diaphragm in the abdominal cavity. So below the diaphragm, the outer layer will be serosa, which includes the mesothelium, simple squamous epithelium, and underlying loose fibrous connective tissue. Now we start with the organs of oral cavity. Most of them were discussed previously in the spring semester. So between the lips and cheeks in frontal part and the teeth, we have the vestibule, and the oral cavity proper is shown here. It is between the teeth, the cheeks, the hard and soft palate, and the floor of oral cavity from which the tongue arises. And now we will look more closer to the structure of the tongue. So here it is shown. It lies on the floor of the oral cavity. It is the muscular organ containing skeletal striated muscle tissue because it is voluntarily regulated. Structurally, it includes the root and the body. In the root, at the surface, we can have the tonsil, the lingual tonsil, which is the large amount of lymphoid tissue to perform the immunity function in the gate of infection, which the oral cavity is. And in the body of the tongue, we have the dorsal surface or the dorsum, the tip or the apex, the lateral surface, and not seen here, lower surface. And here we can see that the surface of the tongue is not flat, it has some elements of relief, which are called the papillae. So if we make the sagittal section of the tongue, here we see the dorsal surface in the upper part, and in the lower part we have the lower portion. We can see the difference in structure. In upper surface, the mucosa is here of specialized type because it has papillae. Such type of mucosa is called specialized because it is specialized for the function of tasting of food, because the papillae, 3 of 4 types, contain taste buds that act as the receptors, which help us to feel the taste of food. Epithelium here is stratified, squamous, partly keratinized, because it is keratinized on the filiform papillae. And here we have no submucosa, and lamina propria mucosa is firmly connected to the muscular body of the tongue. Muscular body of the tongue fills most of the volume of the tongue. It contains muscle fibers. So these long elements here are muscle fibers. They are located in 3 different planes. So somewhere we can see them longitudinally sectioned, somewhere cross-sectioned, and somewhere oblique sectioned. So in the upper surface we have just the mucosa, which contains epithelium and lamina propria mucosa. Lamina muscularis mucosa is absent, and the lamina propria mucosa is firmly attached to the skeletal muscle tissue of the muscular body of the tongue. This firm attachment is needed to produce the function of the chewing, because the tongue helps us to mix and chew the food. In the lower surface, we have the mucosa and submucosa. Submucosa provides motility of the mucosa. Mucosa here contains epithelium and lamina propria mucosa. Lamina muscularis is also absent. Lamina muscularis mucosa is absent everywhere in the organs of the oral cavity. It starts to be present only in esophagus and further in stomach and intestines. Mucosa here on the lower surface of the tongue is of lining type because it contains no papillae, and the epithelium here is stratified, squamous, non-keratinized. Here also in this picture you can see some secretory portions of the glands located between the muscle fibers of the muscular body of the tongue. Their ducts should lead to the surface of the tongue. Here we can see the cross-section of the tongue, also showing the papillae located on the dorsal surface. We can see here the fungiform papillae, which are mushroom-shaped, and the filiform, which have the stratum corneum because they contain keratinized epithelium. In submucosa, submucosa is present only on the on the lower surface, and on the lower surface we have the lining mucosa without any papillae. Most of the volume of the tongue is filled with skeletal muscle tissue. In the lower picture on the left, you can see two kinds of the papillae, which can be found on the dorsal surface of the tongue, which are fungiform and filiform papillae. On the lower surface, the epithelium has no such evaginations, so no elements of relief, and epithelium here is just stratified squamous non-keratinized. Lingual papillae are evaginations of epithelium and lamina propria mucosa. They contain taste buds except of filiform papillae. We have four types of papillae. The largest ones are called circumvallate papillae. They are round shape and they are encircled with this tissue. So they are V-shaped and they are located just between the root of the tongue, which is here, and the body of the tongue. So they are located in this area. Next, second type of papillae are called foliate papillae. They are located on the lateral surfaces of the tongue, like this. Next, and in the most of the body of the tongue, or the dorsal surface, we can see other two types of papillae. The filiform papillae, they are too small to be seen, but they make the white color of the tongue. The white color comes from the exfoliation of the keratinized epithelium. And the fungiform papillae, which are a little bit larger, so they can be seen, they look like small round pink structures between more whitish filiform papillae. So all the papillae except filiform contain taste buds. In the first picture here, we see two types of papillae, filiform and fungiform. They both can be found on the dorsal surface of the tongue. So, as I said previously, every papilla is formed with the epithelium and lamina propria mucosa. They both form this evagination. The lamina propria mucosa forms the primary papilla, which is this larger area, and secondary papillae, which are these smaller evaginations of lamina propria into the epithelium. They enlarge surface of connection between the epithelium and lamina propria mucosa, and this ensures better nutrition of epithelium and better attachment of the epithelium to lamina propria mucosa. So this is the fungiform papilla. Fungiform means mushroom shaped, with the leg and the head. The filiform papilla is shown here. It also has larger primary papilla and these secondary papillae, smaller ones, and the epithelium here is partly keratinized. Next, foliate papillae can be present on the lateral surface. Here we see their primary papillae and secondary should be here. They are smaller. On the lateral surface, in the layer of epithelium, we can see here several taste buds. They are located on the lateral walls of the papillae. And here we see the circumvallate papilla. It also has the taste buds on the lateral surface, and in this area between the surrounding tissue, and the wall of the papilla leads the duct of the special von Ebner gland. This is the protein-producing gland, and the secretion of this gland helps to remove the excess food from this area. When we eat one dish, one meal, we taste it, and then when we eat something different, we want this food, previous food, to be removed from this area, and new food to come here to taste new food. And this happens with the help of this von Ebner's gland. They remove the food from here. Another example of the fungiform and filiform papillae. Fungiform are somewhat elevated upon the surface of the tongue, so we can see them. They have the primary papilla and secondary papillae, and in secondary papillae here it's loose fibrous connective tissue rich in blood capillaries, and here blood capillaries come very close to the surface of these papillae, and this makes pink or red color of them, and we can see them if we look to our tongue in the mirror. In the filiform papilla, we have the partly keratinized epithelium. Another example of the circumvallate papilla, primary papilla, secondary papillae, taste buds on the side wall. The circular groove is the space between the papilla and surrounding tissue, and here into this groove the duct of the von Ebner glands leads. Von Ebner's serous glands. This is our histological preparation showing filiform and fungiform papillae. Our histological preparation is the section of the cat's tongue, and cats have very long and very hard filiform papillae. We can see them here. They help cats to remove meat from the bones when the cats eat their prey. In the human tongue, the filiform papillae are much smaller. They almost are not seen. They are very small whitish structures. They are whitish because of this whitish keratinized epithelium. Between the filiform papillae, we see the fungiform papillae, which look like small round red dots, elevated above the surface of the tongue, and red because of blood capillaries in secondary papillae and non-keratinized epithelium. Another example of these papillae, filiform and fungiform. Circumvallate papilla, surrounded with a groove, von Ebner's glands, their secretory portions and the ducts, which should lead to the bottom of this groove. Foliate papillae, this is actually your picture in the album, because you should draw one such foliate papilla. It has 3 primary papillae, these loose fibrous connective tissue areas, in the form of the crown, 1 in the center and 2 at the sides, and all is covered with the stratified squamous non-keratinized epithelium. In the lateral surface epithelium, we have the taste buds. In the taste buds, the cells are elongated in the direction from the basement membrane towards the surface of the epithelium. Here should be the taste pore into which the food molecules can come to the taste bud and act upon the receptors of the receptory sensory cell. Here we can see such taste buds. They contain several types of cells. Largest one with the larger and lighter nucleus is the sensory cell, others are supportive and basal cells. This is the taste pore. Next, in the tongue we have the lingual salivary glands, which produce saliva. They are simple or compound, and also they can be tubular branched or tubular alveolar branched. Simple and compound are the characteristics of the duct. Here in this picture we can see the compound branched alveolar tubular gland. It means that it has the branched duct. This is why it is called compound, because the simple glands have not branched ducts. This one is branched, blue color is for ducts, so all ducts are branched, and this is why this type of gland will be called compound. And next, into one duct, many secretory portions lead. Secretory portions here are shown in yellow color. If only one secretory portion leads to one duct, in this case the gland will be called non-branched. In our case we have branched gland. It means that several secretory portions lead to one duct. And finally, the last word in the description of the gland describes the shape of the secretory portion. If it is elongated, it is called tubular. If it is sac-shaped or round-shaped, it is called alveolar. If we have both types in one gland, in this case it is called tubular alveolar gland. So, this gland is compound, branched, tubular alveolar. And most of the glands of the tongue are of this type. And depending on the type of secretion, they are also subdivided into mucus-producing and serous glands. In this picture, you can see here the epithelium. Here are foliate papillae, epithelium, lamina propria mucosa, then goes the muscular body of the tongue with the sections of the muscle fibers, and between the bundles of muscle fibers we see the secretory portions and the ducts which lead to the surface of epithelium. Embryonic source of development of the salivary gland. Both epithelium of the ducts and secretory portions derive from ectoderm. So, in glands we have the parenchyma and stroma. The parenchyma here consists of epithelium of both ducts and secretory portions. Stroma is loose fibrous connective tissue between ducts and secretory portions. This loose fibrous connective tissue contains blood vessels, nerves, some immune cells, etc. So, parenchyma, epithelium of secretory portions and ducts, is epithelium derived from ectoderm. The stroma, loose fibrous connective tissue, derives from mesenchyme. According to the type of the secretion, we have the serous glands. They produce protein-rich secretion, but more watery. This type of gland contains serocytes and myoepithelial cells in the secretory portions. In mucous glands, here we see mucocytes and myoepithelial cells in secretory portions. This type of gland produces more viscose, more jelly-like saliva, rich in mucins, glycosaminoglycans. In the lower picture, we see in some areas serous glands, which contain only serocytes and myoepithelial cells. In this area, we see mixed glands. Here we see one secretory portion of such gland. It contains in the central part, mucocytes, which have this non-stained cytoplasm and flattened nucleus moved to the basement membrane, and at the periphery, crest-like, moon crest-like serocytes with their basophilic cytoplasm and round nuclei. Here we see the excretory duct, because here the cells have very small amount of cytoplasm. It means that they are not producing, not secretory cells, because they contain small amount of organelles in the cytoplasm. And we see here two layers of cells, two layered ducts, and this is the lumen of the duct. Between these elements we have here adipocytes and loose fibrous connective tissue. They both will comprise here the stroma. Mucous secretory portions, if we look at high magnification, contain mucocytes with translucent, transparent cytoplasm, which contains secretory granules of mucins, of mucus, and the flattened nucleus moved to the basal portion of the cell. We can see very well the borders between the cells, and the shape of the cells is columnar. Also, we can see very well the lumen of this secretory portion. It's major type of cells in this secretory portion, and second type of cell is myoepithelial cell. It is also cell of epithelial type of tissue, but it is called myoepithelial cell because it contains actin and myosin microfilaments which serve for contraction. These myoepithelial cells kind of hug the secretory portions from the outside. They have long processes and a lot of actin and myosin microfilaments in those processes, and when it is needed, the actin and myosin filaments contract. The processes of myoepithelial cells, which are outside of the secretory portion, contract, and it causes squeeze of the secretory portion, and the secretion is squeezed out from the cells to the lumen and then into the ducts. So, mucous secretory portions produce more viscose saliva, means not watery but jelly-like. Mucocytes produce glycoproteins of the saliva. Mucin is the major type. Between the secretory portions, here we see loose fibrous connective tissue with blood vessels. Here we see serous secretory portions. They contain serocytes and myoepithelial cells. Serocytes have basophilic cytoplasm because the cytoplasm is rich of rough endoplasmic reticulum, the organelle which serves for production of the proteins for the export, which should go out from the cell. The cell nucleus is round in shape. The borders are not very well seen between the cells, and the lumen of secretory portion is also not seen. It is too small to be seen. This type of gland produces more liquid, more watery saliva, which contains water and the proteins. The proteins produced by serocytes include the enzymes amylase, maltase. These are enzymes for digestion of sugars, carbohydrates, and also proteins which are antibacterial, such as lysozyme. Also, the salivary glands produce and release lots of ions, and also the bicarbonate are ions which serve for neutralizing of the pH level in the oral cavity, because the bacteria which colonize our surface of the mucosa and which colonize the surface of teeth, they these bacteria eat the organic molecules from food, and as the result of their metabolism, bacteria produce acids, and these acids will act upon the teeth and demineralize enamel of the teeth, because enamel contains calcium crystals, crystals of hydroxyapatite of calcium, and these crystals can start to dissolve if the pH level drops below the level of 5. So if we have 5 or low pH level, then our teeth will be slowly broken. So the very important function of the salivary gland is to produce bicarbonate carbonate ions, which serve for neutralizing of pH level in the oral cavity and around the teeth, to neutralize the acids produced by bacteria. In this picture we can see both mixed and serous secretory portions and a duct. In the serous secretory portions we can see serocytes and myoepithelial cells. Myoepithelial cells are seen as the flattened nuclei outside of the main mucus or serous producing cells. So these are serocytes with the serous secretory portions. Here we see mixed secretory portions with myoepithelial cells, mucocytes and serocytes at the periphery. And this is the duct, because it contains lining type of epithelium, not glandular as we have in secretory portions, but lining type of epithelium where the cells are not producing any elements of saliva, but it's just the tube for releasing of the saliva. It has 2 layers of cells in its structure, not one. It is related to embryonic source of development. In all glands derived from ectoderm, we will have 2 or more layers of cells in the ducts. In this picture we can see the sagittal section of the tongue. We see its dorsal surface and the lower surface, ventral surface. On the dorsal surface we have both fungiform and filiform papillae. Filiform are covered with keratinized epithelium. This is the lamina propria mucosa, and it is attached to the skeletal muscle fibers of the muscular body of the tongue. In muscular body of the tongue we have different planes of sectioning of the muscle fibers, which go in 3 dimensions. In the lower surface of the tongue we have epithelium, lamina propria mucosa, and then submucosa. We have no lamina muscularis mucosa, which helps us usually to find the border between the mucosa and submucosa, but here it is absent. But here we can see large blood vessels. Large blood vessels are usually found in submucosa. So at the level where we have these large blood vessels, here the submucosa is. Closer to the epithelium there will be the lamina propria mucosa. In muscle muscular body of the tongue, between the bundles of muscle fibers we have adipose tissue, large blood vessels in loose fibrous connective tissue, and also here some nerves and intramural ganglia can be found. The same at high magnification: fungiform papilla, filiform papilla. Inside we have the lamina propria mucosa. It is covered with the epithelium. Lamina propria mucosa is attached to the muscle fibers. And this is the section of the lateral surface of the tongue, because here we can see the foliate papillae, which are present on the lateral surfaces. So, the papillae with taste buds on their lateral sides. Lamina propria mucosa is attached to the muscle fibers of the muscular body of the tongue. Between the bundles of muscle fibers, which are red in color, we have the secretory portions of the serous glands. They are serous because we can see the basophilic cells. These more darker looking with the white lumen inside. These are ducts of these glands. Ducts start at the level of secretory portions and lead to the surface of epithelium. They will lead actually to the groove between the two adjacent foliate papillae. So every duct should lead here, this way. It is seen in this picture. Here you can see how the duct of the gland leads to the area, to the bottom of this area between two adjacent foliate papillae. In this picture, we can see the serous and mucous glands. Mucous glands are seen because we can see here transparent cytoplasm, not stained cytoplasm of the mucous cells. In the same preparation, we can see here the cross sections of the muscle fibers. The myofibrils are seen as red dots on the cross sections, and the nuclei are at the periphery. Between the muscle fibers, we have thin layers of loose fibrous connective tissue. Some sections are cross sections, some are oblique sections. In some areas, we have longitudinal sections in the tongue, because in tongue we have muscle fibers in all three dimensions. Here we see the gland with secretory portions and the duct. The duct contains the lining epithelium. It looks more dark because here the cell nuclei are located more closely together to each other, because the amount of cytoplasm is very small in these cells, because they do not produce anything, they just form this tube, and the lumen is very well seen. The same here, secretory portions and the duct. The same at high magnification, some muscle fibers at longitudinal section, and on the oblique or cross section we can see myofibrils which run along the muscle fiber, and the cross section. These are the nuclei of muscle fibers, which are actually nuclei of the myosymplast and the nuclei of myosatellite cells, which are stem or cambial cells for muscle fibers. Here we see the adipose tissue, the adipocytes with their flattened nuclei located at the periphery. Another example of the serous gland with its serous secretory portions here, and some myoepithelial cells are seen here, the flattened nuclei. And we go to the structure and development of the tooth. This is actually the same as you studied before in the spring semester, so we will take just a brief recall of this information. So, the tooth includes the crown and the root and the neck between them. The neck can be the clinical, it's the site of attachment of the gum epithelium to the enamel of the crown, and the anatomical, it's the border between the enamel of the crown and the cementum of the root. So where the enamel meets cementum, there will be the anatomical neck. It is not seen for the doctor, and the doctor sees this, the clinical cervix. In the center of the tooth we have the pulp. It is covered with the dentin, which is present both in the crown and in the root region, and the outer layers are enamel and cementum in the root region. The cementum is attached to the bone of the alveolus with the help of the periodontal ligament. The enamel is the hardest substance in the body. It contains the calcium hydroxyapatite crystals. It is acellular, has no cells. It is the unique tissue because it contains no cells. Because the cells were alive previously, they produced this material, enamel, and then they died. The structural element of the enamel is called enamel prism or enamel rod. It is elongated structure which runs from the border between the enamel and the dentin towards the surface of the enamel. And in the cross section, every rod is the keyhole shaped with the head round region and the tail region. And every enamel rod consists of elongated hydroxyapatite crystals. These crystals can be seen in this scanning electron microscopy picture. So every this small elongated structure is one hydroxyapatite crystals. So these crystals make both enamel rods and the inter-rod regions, which correspond to the tails of the adjacent rods. In the enamel, about 98% of materials are these hydroxyapatite crystals, and other 2% are actually the water and some proteins, such as amelogenins, ameloblastins, enamelins, taftalins, some of them present in the mature teeth, some of them are removed during the process of mineralization of the enamel, during the maturation of enamel, when the cells, ameloblasts, are still alive. The enamel is covered first from outside with the enamel cuticle, which contains of two layers, the primary cuticle or Nasmyth membrane and the secondary cuticle, the outer layer, which is composed of the remnants of the outer enamel epithelium and the intermediate epithelium. So, in the stage of histogenesis of the tooth, we had the enameloblasts, which make enamel. They produce enamel rods, and finally they end producing enamel with the production of the layer of glycoproteins called Nasmyth membrane, which will cover the enamel rods from the outside. And enameloblasts were covered with the outer enamel epithelium and the stellate reticulum. They come close together and form the secondary cuticle. So this secondary cuticle covers the enamel before the tooth eruption, and after the tooth erupts, the cuticle covering enamel is very fast, very soon removed during chewing, and it stays only in the region of the neck of the tooth, where it helps to connect the enamel to the epithelium of the gum, of the gingiva. And in the mature tooth, the enamel is covered with the enamel pellicle. Enamel pellicle, here we can see the tooth with its two roots. So the surface of enamel covers with the glycoproteins from saliva. These glycoproteins form the film, the thin film upon the surface of the teeth, and this film of glycoproteins, which is called pellicle, protects the enamel of the tooth from the acids, and it lubricates and protects from other demineralizing agents. But also this layer of glycoproteins is the very good environment for bacteria. So when the pellicle is formed, in several hours it is colonized with bacteria, and now the pellicle is transferred to the dental plaque. Dental plaque is seen as this whitish, yellowish substance covering the enamel closer to the gingiva, mostly in this area. So it contains a lot of bacteria. Bacteria eat glycoproteins and release and produce acids. Acids will destruct the enamel. And then, if we do not brush the teeth, this dental plaque grows, and precipitation of minerals from saliva turns the dental plaque into the hardened calculus or tartar. It is now hard because of minerals which are located here, and the tartar or calculus cannot be removed with the brush, so only the doctor can, the dentist, the stomatologist can remove this tartar from the teeth. Now the dentin. Dentin fills most of the volume of the tooth. It is present both in the root and in the crown region. It contains inorganic substances 70% and organic substances, mostly collagen and pre-collagen fibers and other proteins. The dentin is produced by the cells called odontoblasts or dentinoblasts. Unlike ameloblasts, which produce enamel and die after the complete of this process, odontoblasts are still alive in mature tooth, and they are located at the border between the pulp and the dentin, in the outer layer of the pulp here. And their processes go through the thickness of the dentin towards the border between the dentin and enamel, or in the root region towards the border between the dentin and the cementum. Here in this picture we can see the border between the pulp and the dentin. In the pulp, in the peripheral layer, we have the cell bodies of odontoblasts, and in the dentin region we see the dentinal tubules, the holes in dentin, and inside these tubules we have the apical processes of the odontoblasts, called Tombs processes. They run towards the border between the dentin and enamel or cementum. The layers of dentin include predentin, which is more closely to the pulp region. Most of the thickness is the peripulpar dentin, and the outermost thin layer, which is closer to the enamel, is called mantle layer. In these regions, the type and orientation of the fibers differ. Next, dental pulp or pulp chamber. It is the soft tissue inside the tooth. It is composed mainly of loose fibrous connective tissue, and it has three layers. The peripheral layer is called odontoblastic layer here, or L stands for odontoblastic layer. Here we see the cell bodies of odontoblasts. Their processes will go to the dentin, into the dentinal tubules. Next goes the intermediate zone. It is composed of cell-free zone. CZ stands for cell-free zone. In this area we see very few cells. And next, cell-rich zone. CR is for cell-rich zone. Here we see cell bodies of fibroblasts and other cells. And most of the volume of the pulp is the central core zone, with loose fibrous connective tissue, blood vessels, nerves, nerve bundles, and etc. Cementum covers the root of the tooth. We have the acellular and cellular cementum. First, acellular cementum is produced with the help of cementoblasts, which resemble osteoblasts in the bone tissue, and they produce first the organic material, which is then mineralized to form the mineralized cementum. So first acellular cementum is produced, and next in the region of the tip of the root, or in the multi-rooted teeth, also between the roots, there the cementoblasts form the cellular cementum. It means that they do not go to the periphery but produce the cementum extracellular matrix around themselves and become embedded inside the cementum, like osteoblasts transforming into osteocytes in the bone tissue. The same is here. In the cellular cementum, we have cementocytes embedded into the hard extracellular matrix of the cementum. They are located inside lacunae. Their processes go inside the canaliculi to gain the nutrition. And this is in the lower part of the cementum. Supporting tissues of the tooth parodont, they include alveolar bone, the periodontal ligament, and the gingiva. So here we see the connection between the tooth and the gingiva and the bone of the alveolar socket. Enamel is attached to the epithelium of the gingiva with the help of the junctional epithelium between them. The periodontal ligament, which is mainly dense fibrous regular connective tissue, it attaches the cementum of the tooth to the bone and also to the dense fibrous irregular connective tissue of the gingiva. So in the periodontal ligament we have dense fibrous irregular connective tissue and loose fibrous connective tissue with blood vessels and nerves. So periodontal ligaments serves for tooth attachment, tooth support, the bone remodeling because of changing of the strength or changing of the force put upon the alveolar bone, which can start the process of remodeling of this bone. This happens when the brackets are worn by the patient. Also, periodontal ligament serves for proper reception because of the presence of the nerve fibers in loose fibrous connective tissue in it, and it serves for tooth eruption. The vitamin C is essential for periodontal ligament formation and maintenance. If we have the lack of vitamin C in food, then the teeth will not develop in child, or the teeth will fall away, because vitamin C is needed to produce collagen fibers. Without vitamin C, collagen fibers are not produced or not renewed. Embryonic sources of development of the tooth include two sources: ectodermal epithelium, oral cavity epithelium derived from ectoderm, and neuromesenchyme, mesenchyme derived from neural crest. In mature tooth, only enamel is produced from the epithelial ectodermal epithelium, and other parts of the tooth, including dentin, cementum, and the pulp, they all develop from neuromesenchyme. During the development of the tooth, the epithelium of oral cavity and the underlying mesenchyme act upon each other in the form of the embryonic induction. Epithelium develops into enamel organ, which will give us the enamel. Mesenchyme develops into dental papilla, which will give us the dentin and the pulp, and the dental sac, which will give us the cementum and periodontum. Here you see the stages of the tooth development. First stage is the stage of dental lamina, when the epithelium forms this elongated invagination into the underlying mesenchyme along the jaw. Then, second stage, stage of dental bud. From this dental lamina, the dental bud grows, composed also from the same epithelial cells. Next stage is called enamel organ. The dental bud is transformed now into this enamel organ, which contains also of epithelial cells. It is surrounded with a mesenchyme called dental sac, and into it the mesenchyme grows in the form of dental papilla. And next stage will be the histogenesis stage, when the histogenesis of tooth starts. It means the production of the teeth tissues, such as enamel, dentin, cementum, periodontal ligament, and the pulp. So here the inner epithelium of the enamel organ start to differentiate into enameloblasts. The peripheral cells of dental papilla start to differentiate into odontoblasts. The rest of the dental papilla starts to differentiate into the pulp of the tooth. The mesenchyme cells of the dental sac will differentiate into periodontal ligament and cementum. The same shown here. First stage dental lamina, then it is transformed into dental bud. Then dental bud transforms into enamel organ. This stage has two sub-stages, cap stage and bell stage. In the bell stage, the cytodifferentiation starts. Next, histogenesis, and final root formation and teething, because first the crown is formed and only then the root starts to develop. The root starts to develop only when the tooth starts to arise in the oral cavity. Here we see the cross-section of the head of the pig embryo. This is the mouth of this embryo, the tongue, the upper and the lower jaw. In both upper and lower jaw, we have the tooth germs. In the upper jaw, we have the tooth germs at the stage of dental lamina, invagination of epithelium into the underlying mesenchyme. And on the lower jaw, we have the tooth germ on the stage of the enamel organ. In this picture, vice versa, on the upper jaw we have the tooth germ at the stage of the enamel organ, with its inner, outer epithelium, stellate reticulum, its other structures, and on the lower jaw the tooth germ at the stage of dental lamina or growing dental bud. So here we see the tooth development at the stage of the enamel organ. Enamel organ is all this structure. It all is composed of the epithelium, which develops from the epithelium of the oral cavity. So epithelium of oral cavity, the neck of the enamel organ, the outer enamel epithelium, inner enamel epithelium, intermediate enamel epithelium, stellate reticulum, all these structures are epithelium. Around the enamel organ we have the mesenchyme, which is neuromesenchyme. It is here condensed because the cells come closer to each other, not because of movement actually, but because of their very fast proliferation in this area. So here we have the condensed mesenchyme of dental sac, and here dental papilla. Here we see the enamel organ at the cap stage and on the bell stage. On the bell stage, which comes after the cap stage, the enamel organ loses its connection to the oral epithelium and the cytodifferentiation starts. Cytodifferentiation of the inner enamel epithelium means that these cells change into pre-ameloblasts and then ameloblasts. And as the result of the induction, the peripheral cells of the dental papilla turn into pre-odontoblasts and then into odontoblasts. And ameloblasts will next start to produce enamel, and odontoblasts will start to produce dentin. It will be on the next stage, histogenesis. Here, the process of cytodifferentiation at the Bell stage is described. It means how the cells of the inner epithelium turn into pre-ameloblasts, and peripheral cells of the dental papilla turn into pre-odontoblasts. It is described in this slide, and it was described in the lectures on the spring semester, so I will not repeat this information. So here you can see how the pre-odontoblasts start to produce dentin. The Tombs process arises on their apical surface, and the presence of the newly produced dentin blocks the passage of the nutrients and oxygen to pre-ameloblasts. It causes turn of their polarity, change of their polarity, and they start to differentiate into ameloblasts. So here they are ameloblasts, and they start to produce enamel. So odontoblasts are here, they produce dentin. Dentin here is in yellow color. Ameloblasts produce enamel, enamel here in blue color. So the stage when we have ameloblasts and then odontoblasts and they produce enamel and dentin, this stage is called histogenesis. It is seen here. Here we actually see only the tip of the developing crown of the tooth. So in the center we have the dental papilla. It is here the mesenchyme, the peripheral cells of which turned into odontoblasts, and the rest of it will turn into the tooth pulp, loose fibrous connective tissue. Odontoblasts produce pre-dentin first. This is this white area. Pre-dentin is not mineralized organic matrix containing the collagen fibers and other proteins. And then on next stage the mineralization of dentin happens, and dentin becomes mineralized, and here we see this thick reddish pinkish line here. This is the dentin, mineralized dentin. These cells are ameloblasts or enameloblasts. They produce enamel. Enamel here seen as this darker line. Ameloblasts have the Tombs processes seen here. These are the apical processes of these cells through which the proteins and the calcium crystals go from the cell to form the enamel. So here we see the layer of ameloblasts. Then here we see the enamel organ pulp or stellate reticulum, the stellate in shape epithelial cells which serve to provide nutrition for ameloblasts during their life and producing enamel. Here we see blood vessels, and the outer layer here is the outer enamel epithelium. In this region, enamel epithelium comes very close to the stellate reticulum, and intermediate layer, they together will form the cuticle of the tooth. In this slide, the amelogenesis is shown. So first of all, the pre-ameloblasts are cuboidal cells without any processes. They start to produce enamel. First they produce aprismatic enamel, which contains no prisms or rods. Then the Tombs process appear on the surface, apical surface of the ameloblast, and when this process appears, the cell starts to produce rods. When it starts to produce rods, every cell moves in this S-shaped trajectory, S-shaped way, and this causes the S-shaped structure of every enamel rod. So S-shaped enamel rods are due to the way of the enameloblast producing these rods. Last layer will be also aprismatic when the cell is aged. It is aging and dying. It loses its Toms process, produces aprismatic last layer of enamel, and then it degenerates and dies. Here, brown in color, the production of dentin is shown. Dentinoblasts or odontoblasts stay here at the peripheral layer of the pulp. This is the high magnification of the tip of the developing crown of the tooth. Enameloblasts are seen here. Here we see their basal portion. The nuclei are seen in the basal part of the cell, apical portion of the cells, and here Toms processes of the enameloblasts or ameloblasts. So ameloblasts produce enamel. Enamel is seen here as the thin blue line. So ameloblast, next goes the combined intermediate epithelium of the enamel organ, stellate reticulum, and outer epithelium. Here they come close together and they all are very thin and they form here the future cuticle of the tooth. All other cells are the cells of the mesenchyme. Here we see the dental papilla, mesenchyme of the dental papilla, which will turn into loose fibrous connective tissue. The peripheral cells of dental papilla became odontoblasts. Their processes run inside the dentinal tubules seen here. They run from the border between the pulp and the dentin to the border between the dentin and the enamel. So here you see these whitish tubules. They are dentinal tubules containing the Toms processes, apical processes of the odontoblasts. This whitish area between the cell bodies of odontoblasts and dentin is the predentin, yet non-mineralized material of the extracellular matrix, and pink area is the mature mineralized dentin. Here we see the differentiation of the enamel organ, outer enamel epithelium, together with the collapsed stellate reticulum and intermediate epithelium will form the cuticle of the tooth. In the lower portion, the outer epithelium and inner epithelium will form the cervical loop, which will guide the root development. The stellate reticulum, which contains stellate in shape epithelial cells, provides nutrition for ameloblasts. Intermediate epithelium, which is located just outside the ameloblasts, participates in calcification of the enamel and also is the part of the future cuticle. Inner epithelium develops into the ameloblast in the crown region and into the cervical loop and the hairs in the root region. In the papilla, the peripheral cells become odontoblasts, and all other cells will turn into the cells of loose fibrous connective tissue in the tooth pulp. In the area where the root starts to develop, the inner and outer epithelium meet together to form the cervical loop. It elongates downwards and forms the two-layered Hertwig's epithelial root sheath or hairs, which navigates the development of the root of the tooth. Root of the tooth will include the pulp. Pulp develops from the central portion of the dental papilla. Next will go the cementum. Cementum will develop from the peripheral cells of the then goes the dentin. It will form from the peripheral cells of the dental papilla in the area where they meet the cervical loop and the hairs. And the cementum will be formed from the mesenchyme cells of the dental sac. The Hertwig's epithelial root sheet then is broken down, disrupted, and should not be present in mature tooth normally, but can be present and form some small tumors, cysts, etc. So here, this is our histological preparation called tooth development early stage, which is actually the stage of the enamel organ. This is the Merkel cartilage developing bone of the jaw. This is the direct osteogenesis. Mesenchyme is shown here, the enamel organ, with its neck connected to the oral epithelium. Enamel organ contains outer enamel epithelium, inner enamel epithelium, intermediate enamel epithelium, and stellate reticulum. Surrounding mesenchyme is the dental sac, and this mesenchyme is the dental papilla. And second preparation is called tooth development late stage, which is actually the stage of histogenesis of the tooth. This is the oral epithelium. This is the dental lamina. This tooth germ has lost its connection to the dental lamina in this region, but from the same dental lamina in the same region, the germ of the permanent tooth grows, and it will grow in the same bony socket as this milk tooth. So here is this milk tooth. It's developing crown in this region. This will be the future root. It is now very small because it will start to develop when this tooth will be ready to arise, to erupt. So what we see here is the dental papilla, odontoblasts, predentin, dentin, enamel, enameloblasts, future cuticle, which contains outer enamel epithelium, stellate reticulum, and intermediate epithelium. Outer enamel epithelium will meet together with inner epithelium in this region and form the cervical loop, which will navigate the development of the tooth root. And finally, we go to the structure of the esophagus. Esophagus has three anatomical and histologically different portions: the upper, middle, and lower portion, shown also in this picture. In the upper portion, what is the feature and difference from other portions? In the tunica externa, we have only skeletal muscle tissue. So, tunica muscularis externa. Muscularis externa includes here only skeletal muscle tissue. In the middle portion, we will have both skeletal and smooth muscles, and in the lower portion, closer to the stomach, we will have only smooth muscle tissue. This is the drawing, the picture of the cross-section of esophagus at the level of the upper third of the esophagus, because here we can see only skeletal muscle fibers, both in the inner circular and in the outer longitudinal layers of the esophagus. Here you can see the folds, longitudinal folds, which can be seen in anatomical preparations. Folds are the largest types of the relief, made up with the tunica mucosa and submucosa. They both form this folding, this invagination. So we have here the four tunics tunica mucosa, tunica submucosa, tunica muscularis, and externa adventitia, because it contains here no mesothelium. Mesothelium will be present in the tunica externa in the lowest portion of the esophagus, which is located below the diaphragm. This is the diaphragm. This portion, the closest to the stomach, will be covered with mesothelium and have serosa. So, tunica mucosa includes three sublayers or lamina. Lamina epithelia, or just epithelium, it is non-keratinized stratified squamous, then lamina propria, and lamina muscularis. Lamina muscularis here looks like these islets of cells. Lamina muscularis always contains only smooth muscle tissue. Lamina muscularis is absent in oral cavity, and here in the upper third of the esophagus it appears, and it will grow and become larger and larger when we go closer to the stomach. So here we see just the beginning of the lamina muscularis. It contains longitudinally oriented bundles of smooth muscle cells. Longitudinally, it means that they go this way along the esophagus. We have here the cross-section, so every bundle of smooth muscle cells here is cross-sected, and this is why we see here such oval-shaped or round-shaped structures. This structure is the bundle of smooth muscle cell, and each smooth muscle cell here looks like a small circle because they are cross-sected. Next, as I said previously, the border between the mucosa and submucosa is the tunica muscularis mucosa. It is the part of the mucosa. So, just after lamina muscularis mucosa, the submucosa starts. So, this is the start point of the submucosa, and it goes up till the muscularis. It contains loose fibrous connective tissue with some large blood vessels, nerves, nerve plexus, Meissner's plexus, and a large amount of secretory portions of the esophageal glands proper. It means the major type of glands in the esophagus. They are shown here in this green arrow. Their secretory portions are located in submucosa, and the duct leads to the surface of epithelium. It is shown here. Also, in esophagus, we can have these additional glands, which are called cardiac glands, because they are the same in structure and development as in the cardiac portion of the stomach. Cardiac portion of the stomach is this portion of the stomach, which is in contact with the esophagus. And in this region of esophagus, which is the closest to the stomach, we will have these cardiac glands, the same as present in this cardiac region of the stomach. This is why these glands are called so, and also such glands, cardiac glands, can be found at the level of the fifth tracheal ring. These glands are simple tubular. It means that they have non-branched duct and non-branched secretory portions, tubular in shape, and they can contain the same cells that are present in the glands of the stomach. For example, the cells which produce chloride ions, the ions which make the acid in the stomach. So, if we have the same cells producing the acid in the esophagus, it is actually not very good because the surface of the epithelium of esophagus cannot protect itself from the action of acid, unlike the epithelium of the stomach, which produces a lot of mucus and can protect itself from the acid. So, in the areas where we have such glands which can produce acids, we can have the erosions, rupture of epithelium of esophagus. These glands are not shown in this picture. In this picture we have just the esophageal glands proper, which are mucous. The epithelium in the esophagus here is non-keratinized stratified squamous. It derives from the precaudal plate. In this picture you can see the embryo. It has the ectoderm, mesoderm, and endoderm. This is the sagittal section, and actually we see here the ectoderm, endoderm, and in the central line of the embryo we have the notochord, and in the head region, just before the notochord, we have the precaudal plate. And the same is shown here. This is the notochord, and before it we have the precaudal plate. So precaudal plate is the embryonic source of development of the epithelium of esophagus. This is the longitudinal section. So here we see this flattened epithelium, lamina propria mucosa, lamina muscularis mucosa. We can see here the bundles of smooth muscle cells. Next goes the submucosa. Submucosa contains large blood vessels and some nerves and nerve elements. Here we see the esophagus at cross-section. The lumen is not round in shape, it is collapsed, stellate looking. It opens only when we swallow the bolus, the piece of food. So, in normal state without eating, we have this shape of the collapsed lumen and the elements of relief, which are the folds. So here we see the fold, and another fold here, and another fold here. As you remember, the fold contains both mucosa and submucosa, because both these two layers form this shaped invagination. So we have here four layers, four tunics: mucosa, submucosa, muscularis, and adventitia. Mucosa includes epithelium, lamina propria mucosa, lamina muscularis mucosa. Lamina muscularis mucosa looks here like single cross-sected bundles of smooth muscle cells. Then, after lamina muscularis mucosa starts the submucosa. It goes from lamina muscularis mucosa till the muscularis externa. So all this will be the submucosa. It contains smooth muscle, loose fibrous connective tissue with large blood vessels, nerves, lymphoid tissue, and secretory portions of the esophageal glands proper. Here we see their secretory portions, and here the ducts, which lead to the surface of the epithelium. In the muscularis, or muscularis externa, we have two layers, inner circular and outer longitudinal. Externa is here adventitia. The same we see here. Tunica mucosa, from the surface of epithelium till the lamina muscularis. Lamina muscularis is here. It is cross-sected bundles of smooth muscle tissue. After it starts the submucosa. It goes from the lamina muscularis mucosa till the muscularis externa. Here. Then muscularis externa with two layers, inner and outer. Between those layers we have layer of loose fibrous connective tissue with the nerve elements. Here we see the bundles of nerve fibers of the Auerbach's plexus, and the externa adventitia. The same is here. Mucosa from the surface of epithelium till the lamina muscularis, then submucosa, then muscularis. Here we see very well how the secretory portions go into the ducts, and ducts should lead to the surface of the epithelium. And the final histological preparation is the area of transition between the esophagus and the stomach. This is the area where two different epithelia meet together. The epithelium derived from the precordial plate, it is the stratified squamous non-keratinized epithelium of the esophagus, meets with the simple columnar epithelium of the stomach, which derives from endoderm. And this is the weak point for the body, because here epithelial dysplasia can happen and the cancer can develop. And here the type of relief changes. The epithelium of the esophagus is rather flat, as you see here, but in the stomach we have the small types of relief called the pits. The pits are connected with glands. Here we see the lamina propria mucosa, and here lamina muscularis mucosa. Below the lamina muscularis mucosa, the submucosa starts, and in it we have the mucous secretory portions of the esophageal glands proper. As you can see here in this picture, these esophageal glands proper secretory portions can go to the side of the stomach. So here the stomach starts, but in this region we still see the glands of esophagus. Actually, these are the secretory portions, but the ducts will lead to the surface of the esophagus, not to the surface of the stomach. In stomach we have the gastric pits and gastric glands. The glands produce different substance. For example, there we can find some cells producing acids, and these glands are called cardiac glands. Such glands can be found also in the esophagus, in this region, in the region of transition to the stomach, but they are not seen in this picture. If they are present, they are always present inside the mucosa, above the level of the lamina muscularis mucosa. Another example of the same preparation, here the esophagus is on the left and the stomach is on the right. We can see very clearly the border where the stratified epithelium meets with the simple columnar epithelium, and the relief changes from the flattened epithelium of the esophagus without any evaginations or invaginations, and the gastric pits, these pits, invaginations of the epithelium in the stomach. So here we see the gastric pits, which are the invaginations of the epithelium into the lamina propria mucosa, and here some gastric glands. Their secretory portions are located in the lamina propria mucosa. These are the secretory portions of the esophageal glands proper. They are mucus-producing glands. Their secretory portions are located a little bit at the level of the stomach, but the ducts always lead to the surface of the esophagus. The esophageal glands proper are located in the submucosa. It means that they are located below the level of the lamina muscularis. Lamina muscularis mucosa is located above the level of the glands. So here is the submucosa, and here is the tunica muscularis. And one more example of this preparation, the same. The esophagus is on the left and the stomach is on the right. Here we see the lamina muscularis mucosa. It is here. The esophageal glands proper are located in the tunica submucosa, below the level of the lamina muscularis. Lamina muscularis mucosa continues this way here at the level of the stomach. It is located here, and if you look, you see the glands above the lamina muscularis. So here we see the glands of the stomach. They are called gastric glands. And in this region, because this part of the stomach is called cardia or cardiac region, these glands are called cardiac glands. They lead to the bottoms of the gastric pits. So these areas are gastric pits, the areas of the invagination of epithelium into the lamina propria. And the same at high magnification, you see here the area of transition of the esophageal epithelium to the epithelium of the stomach. Lamina propria mucosa, here lamina muscularis mucosa. You see the secretory portions of the esophageal glands proper below the level of the lamina muscularis. It means that they are located in the submucosa. Other glands here are located above the lamina muscularis, inside the lamina propria mucosa. They are cardiac glands of the stomach. The same glands can be found in this region of the esophagus, but they should be present also in lamina propria mucosa. Okay, this was the last slide. So thank you for your attention and see you in class. Goodbye.